Myself is Ashwini Kumar Singh. Today we will discuss about excretory system in human. The excretory system is very important for different types of examinations such as IBU, IGSO, KBPY and different Olympiads. So we will discuss about excretory system in human. The food stuff which is taken by man contain protein, carbohydrates and fats. Minerals and water. During digestion and metabolism, these foodstuffs break into some useful form and some harmful waste materials and liberates the energy. Carbohydrates oxidized and converted into carbon dioxide, water and liberate energy. While fatty acid and glycerol oxidize into carbon dioxide, water and energy. The energy is very useful for our different body activities. When protein converted into amino acid and oxidized, then nitrogenous waste materials are formed. These nitrogenous waste materials are urea, uric acid and creatinine. So we can say the act of removal of waste products from the body is known as excretion. Now, what is the general patterns of nitrogen excretion and its modification? So, general patterns of nitrogen excretion and its modification. Amino acid. As a result of digestion, protein hydrolyzed into amino acid. The excess of amino acid excrete outside of the body, such as UNO, starfish. The second nitrogenous waste material is ammonia. Ammonia is formed as a result of deamination of amino acid. This is the structure of amino acid. Amino acid after deamination and in the presence of oxygen converted into keto acid and ammonia liberate as a waste material, such as the examples of ammonia excreting animal is these are formed in the blood of amphibians, reptiles and fishes. Next is urea. It is synthesized in liver. Urea is synthesized in liver by the help of arginase enzyme. In liver, ornithine converted into citrulline. In the presence of ammonia and carbon dioxide, water is liberated. Citrulline converted into arginine In the presence of ammonia, water is released. Arginine regenerate ornithine and form urea. In the presence of enzyme arginase, urea is waste materials which is eliminated from the body. This cycle is known as urea cycle or ornithine cycle. Ornithine cycle was first discovered by Hans Krebs and Kurt Henslet in 1932. Hence, it is known as Krebs-Henslet cycle. The examples of urea excreting animals are ascaris, earthworm, man, sharks. Next is uric acid. In this case, ammonia is converted into uric acid. For example, reptiles, birds, and insects. Man also excretes some uric acid, which is formed by the breakdown of nucleic acid. Actually, nucleic acid is made up of purine. Purine is composed of two nitrogenous bases, adenine and guanine. Uric acid accumulated in joints of humans and causes a disease, gout. Next excretory waste product is known as guanine. Spiders and some mammals excretes purines. For example, adenine and guanine. Some other excretory waste materials are bile pigments. As we know, the lifespan of RBC is 120 days. After complete their lifespan, RBC break down and its hemoglobin converted into heme and globin portion. So, hemoglobin converted into heme portion and globin portion. Globin is a kind of globulin protein, while heme dissociates into biliverdin. These biliverdin converted into bilirubin. Bilirubin is a type of waste material which is eliminated by liver. The next excretory waste material is carbon dioxide, bile salts, water, vitamins, and nucleic acid. Nucleic acids are made up of purine and pyrimidines. Purines are adenine and guanine, while pyrimidines are thymine, and 5% of total excretion are these substances. In man, urines changed into uric acid, while pyrimidines changed into alanine.